Hi, and welcome to our fifth year revision program. What we're going to do is we're going to do 5 weeks of different topics, and the first topic that was voted this week was cell structure. What we have is we'll look at a few areas relating to it, and then what I'll give you is I'll give you a few questions you can try yourself, and you can practice them at home, and then you can check in the marking scheme how you got on. Okay? If you find this helpful, tell your friends and include them as much as possible. So the chapter we're going to look at today is cell structure, and we're just going to look at three things related to this chapter. We won't do the full chapter in a 15-minute video or so. We'll just look at three things. Okay. First thing we'll look at is the structure of the cell. We'll look at the idea of prokaryotic versus eukaryotic, and then thirdly we'll look at how you would examine a cell under a microscope. So to begin, one of the first things we'll look at is the cells. Okay. So if we take a look at a plant cell, it is more complicated than the cell you would have learned at junior cert. First thing I want you to notice is that the nucleus isn't really labelled, but what it is the nuclear pore. The nuclear pores are the gaps in the nucleus. Okay, the DNA that's within the cell is actually called chromatin. You may have learned the term chromosomes, but actually chromatin is the genetic material that's found inside the nucleus. The outside of the nucleus itself is called the nuclear membrane, and just to notice that there's some gaps in the nucleus that allows materials in and out. Okay. Some differences you see here between plant cells and typical animal cells. First thing is that a plant cell has a cell wall around the outside. Just on the inside of the cell wall, there's a plasma membrane, or sometimes called cell membrane, that's on the inside here. The cytoplasm is the liquidy part that's in between. The vacuole here, the vacuole is the space that's filled with sap. It's also for storage, and it protects the cell. Over on the left here we have mitochondria. Mitochondria relate to respiration. What they do is they make energy for a cell. And that's the same for plants, but it's also the same for us. And also something that you might notice that is there is the chloroplast. Now a chloroplast is not found in a plant cell, which means that, it's not found in an animal cell. A plant cell has a chloroplast, and that means it is able to make its own food by photosynthesis. If we had chloroplasts, we would be able to make our own food, but for plants, they are capable of doing that. So they could ask you to draw a plant cell, or they may ask you to label some of the parts relating to it. It's important to know those functions. It's probably good to have one sentence for each, so even if you wanted a sentence for DNA, you could say contains genetic material. Some of the questions I've set for you this week involve some cells where you have to label the parts and give the functions relating to them. The last one there that we didn't mention is ribosomes. There's lots of ribosomes in the cell, and what the ribosomes do is they make protein. We mentioned the cell wall and cell membrane a minute ago. The cell wall actually gives the plant cell its shape and support, and a cell membrane it controls what enters and leaves a cell. It's kind of like a security guard for a cell. It can allow some materials in and not other materials. Secondly, if we have a look at an animal cell, again more complex than what you would have studied at junior cert, but you can notice a lot of the same materials. Our cells have a nucleus, i.e. with the nuclear pores, the gaps, okay? They've got a nuclear membrane around the outside, and they have some DNA inside in the nucleus as well. The DNA itself actually cannot leave the nucleus, and if you've done the DNA chapter, you should know that from protein synthesis. You may notice some of the other materials that are similar. There is a cytoplasm, there is a plasma or cell membrane, and there's also a number of mitochondrions. One mitochondria is called a mitochondrion, but typically we have lots in the cells, so they're called mitochondria when we have more than one. Finally, the little red dots as well are also, as we mentioned, they're called the ribosomes, and they are where we make protein. We make protein for making new muscles, making new hair, making new enzymes, making new hormones, and the ribosomes are responsible for that. So again, they could ask you what parts are in an animal cell, and they may ask you to compare the differences. So again, I'll just flick through a few of these. Okay. One of the things relating to some of the questions you have this evening is where we look at the structure of a cell membrane. If you were to look at a cell membrane, under a microscope, it would look like this. You actually have two things. You have proteins, and you also have what is called phospholipids. They're the two things that make up cell membranes. You'll also notice there's a pore in the cell membrane as well. These are kind of gaps that allow things in and out of the cell. And your cell membranes are very advanced. They can actually tell whether or not a substance should be brought into the cell, and they can decide whether or not they want to allow them in. Okay. Now, if we're going through a full class, there is some other material that we would look at on this chapter, but I'm just going to pick out a couple of things for you to focus on this week, okay? So we looked at all the parts there, and we mentioned basically their functions in one sentence. Here are some of the differences that you could be asked. A plant cell has a cell wall, whereas an animal cell does not. Plant cells have chloroplasts, whereas animal cells do not. And then finally, plant cells have large vacuoles. And what I've written there is animal cells have no vacuoles, but actually some books would tell you that animal cells actually have small vacuoles. So that might be a better answer to say in that regard. Okay. So that's the first thing, the parts of plant and animal cells and their function. The second thing we'll look at today is two terms, prokaryotic and eukaryotic. If we start with prokaryotic first, these cells do not have a nucleus or membrane enclosed organelle. So what that means is they're basically very simple living things. To have a nucleus is considered, I suppose, complex. So there are some living things that don't have a nucleus or don't have other organelle. By the way, the term organelle there is referring to, mitochondria, ribosomes, chloroplasts, all of the parts that we mentioned previously. And bacteria would be one example. Now, if you remember, there's a chapter on the 5 kingdoms of life. Monera are the kingdom that bacteria fall into, and they would be the example of a prokaryotic organism. What we are is we fall into the group that's called eukaryotic. These are cells that have a nucleus and other cell organelle enclosed by membranes. So, i.e., if you can see there, we have a nuclear membrane, we have a plasma membrane, we have a number of different membranes, and all of our organelle are within the cell. So the ribosomes and mitochondria. It says there humans are eukaryotic, but actually, if you know the 5 kingdoms of life, all the other 4, other than bacteria, are eukaryotic. So that's plants, animals, fungi, and the final one is called protista. So all 4 of those are actually eukaryotic cells. Okay. The last thing then that we'll look at, we won't look at the parts and the functions of the microscope, but instead what we'll talk about is we'll talk about the experiment that you use a microscope, because I've given you a question relating to this. So the first thing there is you place your slide on the stage, you turn on the light source, and you place the clip on the slide to hold it in position. The first one you use the coarse focus knob or coarse focus wheel to place the lens over the slide and move it up and down to focus the slide. You adjust the light by using the condenser and the diaphragm. People often don't know where they are, but the condenser and the diaphragm are actually just below the stage. Okay? And the coarse focus wheel is actually it's the large wheel that's there. The final one then is you should use a fine focus wheel to better focus your slide. So remember that order. You use the coarse focus wheel first, and then you use the fine focus wheel afterwards. Okay. As part of this chapter, you have to be familiar with preparing a plant cell and preparing an animal cell. Now, if we look at plant cell first, we'll look at an onion as an example. So if we looked at an onion cell under a microscope, here are the steps that we would require. So number 1, we could use the forceps, which is like a tweezers. Use the forceps to pull a strip of thin, transparent epidermis from an inner leaf. Basically just take a small, thin piece of onion. Place the small piece on a strip on the slide. Now this is crucial, the stain here is iodine. Add iodine and this stains the nucleus and shows the membranes. Number 4, you add a few drops of water to prevent the cell from drying out. And number 5, you add the cover slip at an angle because it eliminates air bubbles. I don't know if you've ever done this and you've put the cover slip on flat. What happens is you get this big air bubble in the middle of your slide and you can't see anything. So you lower it at an angle to prevent air bubbles. Again, if they ask you a question on this, they could ask you what you did and why, so these steps are quite important. This is what your cell will look like. So you will see the little dot, which is the nucleus, and you'll see the cell wall around the outside. Okay. If we're looking at animal cells, an example of an animal cell would be our cheek cells. So what you can do is you can do a lot of the same procedures. You can use you can use like a cotton swab to take some cheek cells and put them on a slide. Again, you're going to use a stain, and you're going to lower your cover slip at an angle as well. The difference between plant cells and animal cells is that we have to use a different stain for our animal cells, and we use what's called methylene blue. Okay, so remember iodine for onion, and then we use methylene blue for animal cells. And again, you do the same steps that are involved. You put the slide on the stage, you put the clips in place, you start with a low power objective lens for rough focusing, and then use the coarse focus knob, and then you use the fine focus wheel for precise focusing at the end. Just a little note on that, you can't say the coarse focus wheel is used for coarse focusing because you're using the term to explain the term. So just make sure that you say the coarse focus wheel is for rough focusing. Okay. Yeah, so that is essentially what you need to know. Here's the cells here. What you can see is the circle in the middle is the nucleus of the cell, and then the cell membrane is the surrounding parts. Now there's a couple of questions there, but what I've done is I'm going to upload a series of questions for you to try within the fifth year course that we set up. So if you'd like to try them, you can, and the marking scheme is there for you as well. If you have any questions for us, what you can do is you can send us a message on Edmodo, or you can always comment underneath one of the posts, and we'll get back to you with an answer as much as we can. Hopefully you found this helpful. Let us know how it goes, and we will put up a poll for the following week after that. Thanks.